So most of us are pretty familiar with mRNA technology by now, right? But what if you could use it to create a vaccine that's completely personalized, designed for just one person to fight their specific cancer? Well, that's exactly the kind of incredible science we're jumping into today. We're looking at a study that's basically building a new weapon against a really, really tough type of breast cancer.
All right, here's how we're going to break it down. First, we'll talk about the problem, this formidable cancer. Then, the amazing solution, a personalized vaccine. We'll see how it actually trains an immune army, look at what happens when the cancer decides to fight back, and then wrap up with what this could mean for a whole new era of treatment.
Okay, first things first. Let's get to know the opponent we're dealing with here, because this is a particularly aggressive form of breast cancer. So the huge challenge with triple-negative breast cancer, or TNBC, is just how stubborn it is. You can go through treatment, everything can look successful, but it has this nasty habit of coming back. And that risk is highest within the first 3 years. This is the urgent, life-or-death problem that scientists are racing to solve.
So, how on earth do you fight a cancer that's so unique to one person's body? Well, you build a weapon that's just as unique.
The whole process is just brilliantly personal. It all starts when they take a piece of the patient's actual tumor. Then they sequence its DNA, kind of like a detective dusting for fingerprints, to find all the unique genetic mistakes that make it cancer. They pick out up to 20 of the most promising targets. These are called neoantigens. And from there, they build a totally custom mRNA vaccine. You can think of it as creating a personalized, most wanted poster and delivering it straight to your immune system saying, hey, these are the bad guys, go get them.
And get this isn't some sci-fi fantasy that takes years to cook up in a lab. The average time from when they got the tumor sample to having the finished, personalized vaccine in hand, ready for the patient, just 69 days. That's incredible. It shows that this super complex process is actually doable in a real-world hospital setting.
Now, I know this slide looks really technical, but the main thing to see is how cleverly this thing is engineered. This isn't just some basic piece of mRNA. No, it's designed with special add-ons, that cap and signal part you see, that do two absolutely critical jobs. First, they protect the mRNA so it doesn't get shredded on its way into your body. And second, they act like a megaphone, making sure that most wanted message gets blasted loud and clear to all the right immune cells. So the theory is solid, the engineering is smart, but the million-dollar question, right, is always, did it actually work? Let's get to the results.
I mean, did sending out that custom most wanted poster actually lead to a real, trained army of cells ready to fight the cancer?
And wow, this picture says it all. What you're looking at is a test called an ELISA spot assay. On the left, before the vaccine, almost nothing, a few lonely dots. But look at the right side, after the vaccine. It is an explosion of dark purple spots. Every single one of those spots is a T cell that's been activated and is screaming out the chemical signals to go kill cancer cells. It's the literal picture of an immune army being called to action.
And here's the bottom line. Every single patient in this study, all 14 of them, developed a real detectable T cell response to their personalized vaccine, 100%. The training exercise was a complete success across the board.
Okay, but a successful training exercise is one thing. Winning the actual war is something else entirely. Did this immune response actually protect people? This timeline gives us the answer. Each green bar here is a patient who's still relapse-free. And as you can see, for the huge majority, 11 out of 14, it worked. That immune response translated into long-term survival. I mean, look at this. Some of these patients were followed for nearly 6 years, and the cancer never came back.
But great science isn't just about celebrating the wins. You often learn the most from the setbacks. 3 patients did relapse, and digging into why that happened is just as critical.
So, to understand how cancer can pull a disappearing act, we need to know about something called MHC class 1. The easiest way to think about it is like a little flagpole on the surface of all your cells. Its job is to hold up little bits of protein from inside the cell, the flags, for the immune system to check out. If it holds up a cancer flag, the T cells see it and attack. Now check out what happened with one patient. Her original tumor had some of these flagpoles, which is how the immune system could see it. But the cancer that came back, it had evolved. It was smarter. It had gotten rid of almost all of its flagpoles. The cancer cells essentially went into stealth mode, making themselves completely invisible to the very immune army the vaccine had trained to find them. The other 2 relapses tell different stories. For one patient, the immune response to the vaccine was just the weakest of the group to begin with. And for the other, her cancer came back, but it was actually a brand new, genetically different tumor. The vaccine did its job against the first cancer, but it wasn't designed to fight this totally new and unrelated invader.
So, let's put this all together. What's the big takeaway here? This study is really pointing towards what could be a whole new era of cancer treatment.
What's just so amazing is the quality of this immune response. I know this map looks like some wild abstract art, but what it's showing us is that the vaccine didn't just create one type of soldier. It created a diverse, complex army with all kinds of specialists, frontline attackers, long-term memory cells, the whole shebang. And honestly, this might be the most exciting discovery of all. The vaccine didn't just create a temporary flash in the pan. It created what you could call a living drug. It made the killer T cells, sure, the frontline soldiers ready for battle, but it also created a force of stem-like memory T cells. Think of these as a self-renewing reserve army that sticks around for years, providing this constant, regenerating defense against the cancer.
And hey, you don't have to take my word for it. The study's authors really summed it up perfectly. They wrote that their findings demonstrate the feasibility of individualized RNA vaccines and, this is the key part, they document persistence of vaccine-induced functional T cells. In plain English, they proved you can do this and it creates a T cell army that lasts. Which leaves us with one final huge thought. We've just seen a treatment that could be built in weeks that trains a durable, self-renewing living drug inside a person to fight their own unique cancer. So the question isn't just about whether this works for this one type of breast cancer. The real question is this a new blueprint for how we treat cancer itself?